Welcome to all. In this video we will see banana tissue culture standards, popularly known as BTC, for NCS-TCP certification. So banana is one of the important commercial crop and the requirement for tissue culture propagules are raised year by year in India, and almost all the tissue culture production facilities producing tissue culture propagules through micropropagation and supplied the banana tissue culture propagules to the farmers, and it is highly profitable both the tissue culture production facilities as well as farmers, those who are purchasing and planting in their field. Now we will see the requirements for NCS TCP certification of banana tissue culture propagules. The first eligibility requirements for banana tissue culture certifications and source of seed and minimum quality standards for growing of plants inside the greenhouses and polyhouses are briefly explained in this video. Eligibility requirements for banana tissue culture production. All micropropagation and greenhouse facilities with the changing area between the two-door entry. It have provision of lab coats and chapels and must be approved as per standards and guidelines of the tissue culture greenhouse facilities. And the greenhouse must be free of pests and vectors carrying any pathogens, and all the potting or growth media shall be sterilized and water should be treated free of pathogens, and crop hygienic conditions is maintained during micropropagation, potting, planting, irrigating, movement of the use of the equipments, everything should be maintained in good hygiene. And the double door entrance should be insect proof, and the greenhouse equipped with footwear disinfection prior to entering inside the greenhouse, and ventilation should be insect proof for incoming and outgoing airs, and generally Wellington boots or plastic boots are used to enter and work, and change of lab coat is a must prior to entering inside the greenhouse facilities. And the mother plant must be purchased from a notified varieties and confirmed their identity and origin and date of purchase. The virus indexed by sending the samples to the accredited test laboratories to confirm the free of virus in the mother plant. Especially the viruses banana Bunchy Top Virus, popularly known as BBTV, cucumber mosaic virus or CMV, banana Bract Mosaic Virus BBMV, and banana streak virus BSV are virus indexed and virus free plants are used. In addition, free of endophytic or epiphytic bacteria and fungi also used. And subculture or submultiplication materials need to be obtained from afresh medium of the nodal organization, and once permitted the number of passages of shoot multiplication with old culture has been completed. All the stocks must have a valid varietal identification and a disease testing report at any time and have to keep at least 1 year. The source of seed material or propagule material. The recognized aseptic initiation and propagation procedure should be followed to handle the seedling materials, and initiating facility must maintain record of varietal identification, date of initiation of the culture, origin and testing of the results from the ATL, and have to keep at least 1 year. And for testing virus indexing genetic purity, a minimum of 10 plants or 0.1% population of plantlets for each varieties are selected for testing, and these has to be taken randomly and subject to a minimum 8 passages. Only 8 passages are allowed for mass multiplication, and virus indexed and free of endophytic or epiphytic bacteria fungi materials are preferable. The valid pathogen testing results are required at the second and third subculture stage prior to the bulking up of the culture as a mother plant for further passages. Here we will see the flow chart of NCS TCP guidelines for the production of tissue culture banana propagules. First we have to select the superior clone having superior features like high yielding, duration and good quality, etc. And we have to send it virus indexing and DNA profiling by taking a 0.5 to 1 gram of tissue sample or as per the requirements requested from the accredited test laboratories. Once it is DNA profiling is obtained and free of viruses, we have to process the explant for in vitro initial establishment culture, that is called in vitro culture initiation, and we have to go for 2 or 3 subcultures. Now we will get some more amount of propagules, and again samples are taken and sent for virus indexing and make sure it is free from banana specific viruses. Now this material is virus free certified material and used as a mother stock, and we have to continue subculture in shoot multiplication medium for rapid proliferation of shoots up to 8 passages. So we will get huge number of micro shoots at this stage. Then the micro shoots are separated and placed in rooting medium under aseptic conditions and wait until 2 to 3 healthy roots are formed. Once it is formed, the full plants are carefully taken out along with the culture vessel, gently washed in the tap water, remove any agar medium, and planted in the potting soil mix and sent for acclimatization and hardening purpose. So we can use well equipped greenhouse facilities, shade net or polyhouse for the hardening as well as acclimatization process. Generally it will take up to 6 weeks for hardening and acclimatization, so that the plants can learn how to survive in the field condition. Now the plantlets transfer to nursery, which are kept in a normal shade net, and samples are taken, either 0.1% of plants or 10 minimum plants, and test for virus indexing and genetic fidelity. Once everything is confirmed, now the plantlets are ready for dispatching to field planting in the farmer's field. Next we will see the minimum quality standards for growing of plants inside the greenhouses or polyhouses. The sanitation practices such as free of insect diseases and dust must be strictly adhered, and no field produced banana can be grown inside the protected environment to produce the basic stock, and the varieties must be separated by physical barriers such as proper tagging or barriers. Before field dispatch, the propagule should be virus indexed and tested for clonal uniformity from the sample size of 0.1% plants or 10 minimum. And if the accredited test laboratories identified presence of any pathogens, no material should be sent for dispatching and or hardening purpose. Entire material should be destroyed, and the laboratory or agency producing the tissue culture material should issue a certificate to the banana tissue culture certification agencies have been produced as per guidelines. So once the accredited test laboratory is satisfied and they are issuing the label for banana tissue culture, it is considered as certification. The banana tissue culture label should be supplied in containers with 12 into 6 cm label. The label is signatured with seal containing the name, designation of the concerned agencies. The color of the label is diagonally yellow and opal green, and banana tissue culture producing agencies shall maintain the accounts of labels printed and issued to the dispatch purpose. Thank you.